Okay, so I'm going to try and explain the light dependent stage, or I might as well just do the whole photosynthesis really quickly. So yeah, we'll start with the chloroplast. So I'll just pause the video and I'm going to draw the diagram of the chloroplast, just a brief one, so you can just understand the key principle. So I've drawn the chloroplast here. Obviously, this diagram doesn't show all the organelles within the chloroplast. There's obviously you're going to have different things like this. I don't know, like ribosomes and lipids and stuff. But what I've just drawn here are thylakoids. So I'm just going to label this as that. Again, excuse the handwriting. And they're arranged in stacks called grana, granum for singular. Before I rub this out, just quickly, the space within the chloroplast is called the stroma. So this is going to be really important in the light dependent stage here. So yeah, you just need to remember these. And specifically, the light dependent stage occurs within the thylakoids over here, which I'm going to go into more detail about. But before that, I just want to explain that within the thylakoids there are pigments such as chlorophyll A, B, and you also have accessory pigments such as xanthophylls here, and their role is just to absorb light energy. So yeah, I'm going to go into detail about how then this light energy is used for photosynthesis. Okay, so what I've drawn here is basically the thylakoid membrane. So the one that I showed in the last diagram in the chlorophyll, in the chloroplasts. The ones that made the stacks called the grana. So this is just the membrane of the thylakoid, and I'm just going to label here. So this end of the thylakoid is going to be the stroma, and of course there's going to be the thylakoid itself. Yeah, once again, excuse the handwriting. And of course this in itself is the thylakoid membrane. So what we need to first understand is what I've drawn here. So this thing over here, this is known as photosystem, and this is photosystem 2. And this one is photosystem 1. The reason this is called 2 is because it was discovered later than photosystem 1. So by order of chronology, this is obviously going to be 2, although it's confusing, admittedly. So within these photosystems, you have the pigments that absorb the light energy and allow for the light dependency to happen. So this is in both photosystem 1 and 2. But we're going to start with photosystem 2 because that's where it starts from. And in photosystem 2, you have these electrons in photosystem 2. And I'm just going to rub this out for space. So you have these electrons, and of course you have light energy coming in here. I'm just going to say LE. Again, it's not very clear. I'm going to use a different color. Let's use black. So you have light energy coming here, LE. And what this does, over here, it excites these electrons. So these electrons, they're going to increase in energy. They're going to move into an excited state. So the energy that the electrons contain is going to be higher. And what this does now is it moves these electrons into a high energy state, and they're going to move into something called the electron transport chain, otherwise the ETC. But before we carry on here, we also need to notice the fact that the photosystem here, photosystem 2, has just lost 2 electrons. So to replenish these electrons, it undergoes something called photolysis, or photolysis, I suppose, depends how you pronounce it, which is basically the breakdown of water. So before I carry on further, from GCSE, you remember that photosynthesis is literally just CO2 plus H2O to produce C6H12O6 plus O2, and then to balance it 666. So to put it simply, water and carbon dioxide are the reactants, and oxygen and glucose are the products. So I just mentioned that the photosystem 2 here undergoes photolysis to break down water. So you have water here, and using the light energy that just came from whatever the source is going to allow it to break down into H plus ions. I'll balance in a moment. I know you're very picky about chemistry and stuff, but I'll balance in a moment. It's also going to produce oxygen and also electrons that will now replace this. So to balance, you've got 2 H plus, half O2, and 2 electrons. And you can see we've replenished 2 electrons here with the ones that I've just moved into the electron transport chain. So these electrons are simply just going to go into the photosystem 2. And these H plus ions will just stay here in the thylakoid side. So you can see how we've used one of the reactants, which is H2O, to produce one of the products, which is a waste product in this instance, O2, over here, which in a plant will just simply diffuse out through the stomata or wherever. So key idea you need to know about photosystem 2 is that it undergoes photolysis to break down water to mainly replenish the electrons that are lost into the electron transport chain by the breakdown of water. So I just mentioned how H plus ions were also produced here. So I'm also going to draw these H plus ions here. And this is in the thylakoid space, by the way. Remember, this side is the stroma, which is outside the thylakoid, and then this is within the thylakoid, and the membrane is the one separating them. So we've got H plus ions here in the thylakoid space. So going back to the photosystem. So now we've had electrons moving into the electron transport chain, and they're going to move down the electron transport chain into photosystem 1. But what this does, the movement of electrons down the electron transport chain produces ATP by phosphorylizing an ADP molecule because electrons are moving down the electron transport chain. So this will produce ATP. Another thing you need to know is the fact that there are also H plus ions roaming around in the stroma. And so obviously this over here is producing ATP. So we can just say in this diagram that this is just producing ATP. And now this ATP is used to actively transport these hydrogen ions across the thylakoid membrane into the thylakoid space. So now you've got a lot of H plus ions here. And yeah, so this creates a concentration gradient between the stroma over on this side and the thylakoid space on this side. So naturally, the H plus have a tendency, so obviously there are less H plus now, there's a greater concentration of H plus here. They have a tendency to move back into here by passive transport, which is diffusion here. They do this through the ATP synthase molecule over here. So when H plus ions move through this ATP synthase molecule, this produces ATP, which is one of the things that we'll need for the Krebs cycle. But we're not done quite yet because now we have electrons here in photosystem 1. So this is like the first part of the light-dependent stage, to produce ATP through the ATP synthase molecules by moving H plus through the ATP synthase molecule to produce ATP, which is one of the things that, as I said before, we need for Krebs cycle. So now I'm just going to rub this out. And we can move to the second part of the light-dependent stage, which happens in this photosystem 1 area. So now photosystem 1 has got these electrons, these ones, that have moved down the electron transport chain into photosystem 1. And now these electrons go down an even shorter electron transport chain, to which these electrons are then picked up by a co-transport, a coenzyme called NADP+. So now I'm just going to move this down here. The NADP+ picks up 2 electrons, but also remember that there are loads of H plus ions roaming around now here through photosystem 2's H pluses going through the ATP synthase molecule due to facilitated diffusion. So now there are loads of H plus roaming around here, which are now going to be picked up by NADP+. So one H plus ion, which produces something called reduced NADP or NADPH, and this is a product that we're going to use in the light independent stage or the Krebs cycle. So to summarize, I'll just put it into words. Basically, light energy here activates or excites the electrons here in the photosystem 2, which allows them to move into a high energy state in the electron transport chain. They move down the electron transport chain, which produces ATP. Now this ATP allows H plus ions to be actively transported into the thylakoid space from the stroma through the thylakoid membrane over here. I'm not sure if you can see my circle here, but I'm just going to zoom you can. So they move them into the thylakoid space, and now you have a buildup of H plus or protons here. So ATP synthase allows the H plus to move by facilitated diffusion back into the stroma to reach equilibrium through diffusion, which is a passive style of transport. So this produces ATP, which is one of the products that we need. And at the same time, as we said, electron transport chain moves electrons into the photosystem 1. And then in photosystem 1, the electrons also get excited and move up a shorter electron transport chain, to which they are accepted by this NADP+. And the NADP+ also accepts hydrogen ions, or H plus ions or protons, and they bind together to form NADPH. So that is the light-dependent reactions. Oh, and one more thing that I forgot to mention. Obviously, when this moves down the electron transport chain from photosystem 1, it also produces ATP, and when the NADP+ accepts the the electrons, this is called non-cyclic phosphorylation. However, the electrons don't necessarily have to be picked up by NADP+. They can just return back. So suppose you have photosystem 1 again. Sorry for the handwriting. Once they move down the electron transport chain, they obviously release ATP, and they can just return back to photosystem 1, and this can continue over and over again. And this is called cyclic phosphorylation, because you're phosphorylating an ADP molecule to produce ATP, which can be used for energy. So yeah, so this is called cyclic phosphorylation. But for the light independent stage, you do need NADPH or reduced NAD for the Krebs cycle, which I'll move on to now. Okay, so we've got Krebs cycle. So I'm just going to reiterate the formula that we used at the beginning for photosynthesis. So it was 6CO2 plus 6H2O goes to make C6H12O6 plus 6O2. So you will have noticed that we've used H2O and we've produced O2, which was a waste product, and this was obviously used in photolysis. But we haven't used CO2 and we haven't produced glucose in this case yet, which is going to be completed by the light independent stage, which is the Krebs cycle. So immediately, one of our reactants is going to be CO2, and I'm also going to put the number of carbons here to make it easy. Oh, hold on, sorry. Oh, hold on, sorry. I made a mistake. This isn't Krebs. Krebs is for respiration. This is Kelvin's. I've been saying Krebs this whole time. It's Kelvin cycle. Yeah, apologies for that. And it's important to know that the light independent stage or Kelvin cycle, I think happens in the stroma. Which, if you remember the chloroplast, this is the thylakoid. What I just drew, by the way, above is the thylakoid membrane between here. This space is here, known as the stroma, and of course these things in stacks are called thylakoid. Yeah. So the first part of the Kelvin cycle, as I mentioned before, you start with CO2. So CO2 will diffuse into the stroma of the chloroplast, where it's met with an enzyme called, it's met with an enzyme called rubisco. But this enzyme is used to catalyze a reaction between CO2 and something called ribulose bisphosphate, which is a 5-carbon compound. Oh yeah, as I said, I'm going to write the carbon chains over here. So this is 1-carbon compound. And we have a 5-carbon compound called ribulose bisphosphate, which I'll just write as RUBP, which you can also write as, which is a 5-carbon chain. And this reacts with CO2 and is catalyzed by rubisco. I'll draw the arrow again because it's more convenient. The CO2 also comes in here and binds with ribulose bisphosphate, or RUBP. And this produces, obviously, 5 + 1, a 6-carbon compound chain, which we'll just call an intermediate compound for now. Intermediate compound, which is a 6-carbon chain. Oh, and by the way, the reaction between ribulose bisphosphate and carbon dioxide is known as carbon fixation, because you're adding a carbon onto a 5-carbon chain to produce 6-carbon. So this intermediate compound right now is unstable, so it will break down to form 2 lots of a 3-carbon compound chain called glycerate 3-phosphate, I think. Glycerate 3-phosphate, but you can call it GP as well. I think they call it that in the textbooks as well, so no biggie. And this is a 3-carbon compound, and this will then produce. This is also unstable, so we'll produce a triose phosphate. 2-TP, you can call it, and this is going to be 3-carbon compound as well, because triose is 3. And obviously 2 lots of this makes 6 carbons. But now you'll notice that we're going from 6 carbons to 5 carbons, and you'll be wondering, where's this extra carbon gone? That's a valid question. And this carbon has gone elsewhere to make other carbohydrates such as glucose. So you've lost 1 carbon, and you'll notice from our equation for photosynthesis, C6H12, it's a 6-carbon compound. So this Kelvin cycle occurs 6 times to produce 1 molecule of glucose, because you're releasing 1 carbon each time per cycle. So now we'll go into more detail about this cycle now. So yeah, we mentioned rubisco as the enzyme for this. And from the light dependent stage, we have reduced NAD pH. Remember, H is signifying that it's got a hydrogen, therefore it's reduced. And this is going to produce NAD P, simply. And this process, I'm going to roll this out because I need more space for this. This process also uses ATP to turn glycerate 3-phosphate into triose phosphate. It's going to require ATP as well. So NAD pH turns into NAD P, NAD P plus ideally. And we also produce lots of ATP from photosystem 2 side of the light dependent reaction. So this is going to produce ADP plus PI, phosphate group. And finally, to turn this triose phosphate into ribulose phosphate, you also need another ATP molecule. So you can see how the products of the light dependent stage are being used here in the Kelvin cycle. So overall, this is the Kelvin cycle. Apologies for the horrendous diagram, but hopefully you can sort of see how it works here. So to summarize once again, you've got a 1-carbon chain in carbon dioxide, which binds with this ribulose bisphosphate, which is 5 carbons. So 5 + 1 makes 6 carbons, which is the intermediate compound. This is known as carbon fixation and is catalyzed by rubisco in the stroma. This intermediate compound is unstable, so we'll split into 2 glycerate 3-phosphate groups, or 2GP, which are 3-carbon compounds each per molecule. And you use the products from the light dependent stage to turn the reduced NADP into NADP+, and you also use ATP to turn the GP into TP. And once again, you use ATP to turn the 2 triose phosphates into ribulose bisphosphate. But at the same time, since you're losing a carbon here, this carbon is going away to form a carbohydrate or any compound. It could also be lipid or an amino acid, whatever. So yeah, so I'm just going to refer back to the equation. So you have CO2 plus H2O 6 goes to form C6H12O6 plus 6O2. So you can see we've used CO2 here as one of our reactants. Done. This was used in photolysis. This is being produced over here, and this will be done after this has completed 6 cycles. So we have produced this, and O2 was a waste product in photolysis also. So yeah, I think that covers it all. Apologies for the long video. Of course, I would recommend speeding it up. I'll try and edit it to speed it up, but if I don't get the time, I think I'll just send the raw video to you. But yeah, so light dependent stage and light independent stage. Cool.